Thank you very much for the invitation and the opportunity to share our work here with you. A VISTA system really provides neuroscientists a sophisticated approach to visualize real-time cell dynamics, causing imaging in the freely moving animals. And most early adopters of this system are actually using this system to address the fundamental questions like how neural circuits shape behaviors. So my lab is actually using this system from a very different perspective. We're using this system to visualize migration dynamics of newborn cells in the adult hippocampus. So in the next few slides, I'm just going to give you a brief introduction of the brain area of our interest and why we're studying neuromigration. So my lab is interested in activity-dependent mechanism underlying adult hippocampal neurogenesis regulation. And as you may or may not know, under physiological conditions, there are two neurogenic regions in the adult mammalian brains. So they are, one of them is the lateral ventricles in the free brain. So where neurons arise from subventricular zone and moving to the olfactory bulb. So the other neurogenic region is the dentate gyrus in the hippocampus. So where cells are generated locally within a region called the subgranular zone. So this is the region of our research is based on. So adult hippocampal neurogenesis comprises multiple distinct developmental stages. Upon a neurogenic cell division, stem cells gave rise to intermediate progenitor cells, which in turn gave rise to proliferating neuroblasts and post-mitotic immature neurons, and finally mature dentate granule cells, which are integrated into the existing circuits. So strikingly, those post-mitotic neurons within a clone, they're really well positioned. They're perfectly spaced out. So each cell, so whenever I'm talking about a clone, that means that all these cells are actually derived from a common progenitor cell. So you can see all these cells really occupy their own territory and extend their dendrites and sending their axonal projections. So why newborn cells can really occupy their own positions? So it is known that appropriate positioning of the newborn cells is through an active process of migration from the site of origin to the site of targeting locations. So that's the location they want to survive and integrate into the circuits. So that means this is a really fundamental process we're studying here that actually determine the final allocation of the neurons in the neural system. So in general, there are two major types of neural migration predominant during brain development. So they are radial and tangential. So in the developing cortex, excitatory principal cells migrate radially along this radial process of radial glia cells, while the GABAergic interneurons migrate tangentially independent of radial processes. In the adult brain, there are also neuronal migrations going on. So the most well-documented neural migration occurs in the lateral ventricles, as I pointed before. So where cells generated from subventricular zone and migrate to olfactory bulb through a distinct structure called a rostral migratory stream. So when cells migrate to olfactory bulb, they become post-mitotic, and they continue to migrate radially into granule cell layer to become mature neurons. And in the adult dentate gyrus within the hippocampus, it is well established, and this is the only aspect people have studied, that is the radial migration of post-mitotic neurons. So they know the neurons generated along the SGZ region, the subgranular zone, and they move into granule cell layer during their maturation. So the question is, before they become post-mitotic and they migrate into the granule cell layer, are they also tangentially migrating like what they do in the subventricular zone? So this is just something I want to show you here. I think it's a point really worth noting, that is, if cells are doing radial migration in dentate gyrus region, the cells can only move very limited distance because the thickness of the granule cell layer is only 50 micron. But if cells were doing tangential migration, they actually have a really good and big space to work on. So we hypothesize this tangential migration might actually serve as the predominant mechanism to allocate the neurons in the nervous system. But this aspect has not been studied, largely due to the technical limitations associated with tracking these cells. So to solve this problem, and a graduate student, a very talented graduate student, Andrew Crozer, in my lab, and employed this in vivo microendoscopic system in conjunction with fluorescence labeling. And he's implanting all this mini microscope into the hippocampus, which is just above the dentate gyrus of our interest. So he implanted this 1 millimeter green lens here, and this is the field of view that he can see upon LED light on. Basically, we have the lights off all the time. Most of the time, every minute we will turn it on for 1 second, and then we can have, when we gather all these frames together, then we can have a time-lapse video for cell migration dynamics. So using this system, that the questions we would like to address is, does tangential migration really occur prior to the post-mitotic stage? And if so, if this occur during a specific developmental stage? And also, how newborn neurons migrate in the neurogenic niche? So do they have characteristic of velocity, trajectory, and directionality? And then we would like to address the in vivo dynamics of those migrating cells. And also, more importantly, adult neurogenesis is highly activity dependent and experience dependent. So we want to ask if the tangential migration of newborn cells is also experience dependent. And if so, what's the functional consequence of that? And so the first question, as I told you, adult neurogenesis is a process containing multiple developmental stages. So along this lineage, there are multiple cell types involved as well. So at first, we want to look at the cell types really early in the lineage. Those are the most primitive cells in the lineage. So those are the type 1 and type 2A cells. We take advantage of this nestin GFP line. So this mouse line has nestin promoter there, which actually label both radial stem cells and non-radial progenitor cells. So those are the cells here and here. So just a little bit background information for those cell types. So the radial stem cells is considered as quiescent, the most primitive stem cell population. They don't actually express cell proliferation marker because they largely stay quiescent. While the non-radial progenitor cells, most of them actually express a proliferation marker. This is MCM2 here. This is just the 2 cell population we're talking about here. So we actually traced the nestin GFP mice for 8 hours in this video, and we want to ask if those cell is the population that is mobile or stationary. So this is the time frame, this is 8 hours. You don't actually see them really moving a lot. So all these dots, actually, we mark those dots as each the cell body we can see. Actually, there are a lot more, but I think this is already a lot there to keep your eyes busy. So we actually, the conclusion from this is we didn't actually observe significant tangential migration of those cells. And next, we think that, so this neuronal migration must happen in a later stage. So next we look at the type 2B cells. Those are the intermediate progenitor cells. And we use a retroviral based approach. So retrovirus labels dividing cells. So not only the retrovirus expressing GFP can label the cells, but also they can really birth date those cells. So for example, 4 days after retroviral injection, those cells will be 4 days of cell age. So this is what we see from the whole mount from 2-photon microscope. So 4 days after retroviral injection, we see this nice distribution of the cells there. So we hypothesize those cells probably have undergone tangential migration. So we actually did endoscopic imaging on those cells. So here's, I just want to show you the quantification first. So we actually identify there's a peak migration time frame, which is ranging from 3 to 6 days. And when cells older than 10 days, they don't seem to migrate anymore. So here are the video that we get. So you can see this line dots, and you can see they move a significant distance here. So this is about like 5 hours recording. And then some cells definitely move faster and a longer distance than others. And for the cells older than 10 days, they don't seem to migrate. So you can see this 15 days old neuron, and they stay the same place, and we record them again after 3 months, and then they still stay in the same place. So in summary, all this data I showed you really identify a migratory phase, which is during the neuroblast stage, and type 1A and type 2A progenitors do not move, and later post-mitotic immature neurons do not seem to move tangentially. So the second question is, we want to really characterize their velocity, trajectory, and directionality. So what you can see here is we actually can really nicely measure their velocity. We identify their velocity actually peak at 4 days, and then they stay at this peak for several days. And their trajectories are highly variable. Here I have a video that you can really appreciate how variable they are along time frame. So you can see some of them really extend fast. Some of them are actually just extend a really small distance. But overall, that's what you got. So all these dots is their final location. We have all the cells put together as the one starting point. So the other aspect that we can get from this is the directionality ratio, and we also call this directional persistence. So how persistent they are to one direction. So this is in highly contrast to very directed migration in SVZ neuroblast, the olfactory bulb neuroblast. So they are actually have really highly directed migration from the site of their origin, which is SVZ, all the way to olfactory bulb. They don't seem to migrate in multiple directions like this. So we measure their ratio, and you can see like the small d represent actually the direct distance between start to end points, and the big D is the actual distance they traveled. So if the ratio is turning to 1, that means those cells are highly directed. But if their ratio is turning to 0, that means they are actually not very persistent. So here we just showed 2 examples. Is relatively high directional persistence. This is a low directional persistence. So the next question that we want to ask is what kind of in vivo dynamics we can get from those migrating cells. So first, we can actually really use this system to identify a stereotypic migration cycle associated with those cells. So one migration cycle involved in the leading process, extension process, and the nuclear translocation. We also call this process as nucleokinesis. So that's you see like these cells quickly move from this side to this side. Let's play again. So this cell, in comparison, is staying in this place, it's not moving. What we get from this video is, this is the breakdown of the video. Then you can really appreciate how far this cell can move from one position to the other through this nucleokinesis process. So the cell, so average of 2 hours is how they do nucleokinesis. So from leading process extension to they transfer the whole nucleus to a new location. This is average 2 hours. It's slightly variable, but not by much. So the other feature that is very interesting is those cells do not really constitutively just moving all the time, one nucleokinesis after the other. They actually rest in between nucleokinesis. But this time, we call it the idling phase, is actually highly variable. So some cells do not really need a lot of rest, but other cells really need over like 6 hours resting time before they can get ready for the next nucleokinesis. And their mean migration velocity is actually really big during nucleokinesis phase, but not too much in the idling phase. They pretty much just watching and deciding where they want to go. And this is also shown by this cumulative distance. If you give them 1 hour, if you trace nucleokinesis phase, and the cells actually move, the distance of the cell moving increasing a long time, but not too much for idling phase. So together, this data really showed nucleokinesis. It represents a highly mobile phase, which really contributes to the major movement of the neuroblast during tangential migration. So the next in vivo dynamics that we can get from this system is the cell-cell interaction. So the cell is not just moving by themselves. They actually interact with each other. And this could be a migration force for the cells to actually move forward or decide where to go. So what I want to show you here is this video. So it's placed very quick, but what you can get is when they have minimum distance there, one cell starts to push the other cell away. So here is the quantification. So along the time points and what their distance start to increase. So before I move to the next slide, I want to show you a whole picture of this movie to have you appreciate how dynamic the interaction is. So you can see actually they spend a lot of time to interact with each other. We call this a homotypic interaction. And once they have this interaction and one cell start to take action, actually both cells start to take action. It's just one cell take more action than the other. So the cell decide to move away from this cell. So that actually, we think that this must be, so when cells interact, there got to be a lot of cues there. Could be chemoattractive cues, could be chemorepulsive cues, and also motogenic cues that in combination actually constituted with the cell migration. But when they decide to move, the chemorepulsive cues actually take a major role to push the cells away. So the other thing that we can get from this system is because during this phase, cells are highly proliferative, so which you can see here. Cells, the DCX, which is a marker for cell migration, and most of the DCX cells, migrating cells express proliferation marker. So this migration and proliferation has a nice correlation here. Then the question we want to ask is, what's the cell dynamics associated with proliferation? This is the migrating cells, and the mother cell is divide and become two daughter cells, and two daughter cells just move away from each other. This is the breakdown of the cells. So you can see this one big mother cells and go through cytokinesis and become two daughter cells, and these two daughter cells move away from each other. And this chemograph also shows the fluorescence split along the time. And here is just the quantification for relative cell surface area. and the distance between two daughter cells along different time points. Then it comes to the last question we want to address here, that is, if this process is activity dependent, is experience dependent. So one hallmark of adult neurogenesis is their dynamic regulation by animals' experiences. Previously, during my postdoc, and we discovered enriched environment serve as a critical environmental cue to regulate proliferating neuroblast survival. So since this is also the cell type we're interested here, and they are the cell type which are moving, and we wonder if enriched environment actually exert an effect on cell migration. So we subjected animals to enriched environment for 2 weeks before we do retroviral injection and the lens implantation for imaging. And interestingly, what we see here is in a field of view, and a significant higher percentage of cells migrate compared to the normal housing condition. And also enriched environment seem to increase the exploration of these migrating cells. And you can see this 2 graphs here, and the enriched environment really actually seem to increase the cell motility and the cell migration through here. So we actually quantify them through the MSD, and this is the mean square displacement, and this is the quantification that can actually quantify the exploring area. And also, this increased exploration could be due to increased exploration, could be due to increased migration velocity, or directional persistence. So they could move faster, could be due to they're more persistent. So here, what I show the video here is a set comparison of the 2 conditions. Then you can really see enriched environment actually can increase the cell migration distance, cell migration velocity, but not too much on the directional persistence, because under both conditions, cells still kind of moving randomly. So this increased migration velocity could be due to increased time spent in the nucleokinesis phase, as I told you before, because this represents a highly mobile phase. Or this could be due to increased velocity during the nucleokinesis phase. So our data showed it's actually this is because of the former, but not the latter. So the enriched environment increased time of the cells spent in nucleokinesis phase, at the same time reduced the idling time in between nucleokinesis, but not the mean velocity during the nucleokinesis phase. So what's the functional consequence of increased exploration of the cells? We hypothesize the increased exploration of migrating cells could really allow proliferating neuroblasts to migrate further, so that it creates more space between neighboring cells. So to measure the distance between neighboring cells, we took an inducible genetic approach. We use this AICL cell pre-ER line and cross with a reporter line, so that we can label neuroprecursor cells. And what we got, I don't want to get into too much details of the system, but then what you can get from there is we labeled all these neuroblasts, then we can measure the distance between the neighboring cells. So consistent with our hypothesis, that is enriched environment to really actually increase the distance between neighboring neuroblasts. So here is the quantification. So in summary, what I have just told you is there is significant tangential migration at neuroblast stage in the adult hippocampus. So this has been an uncharted research area. Now we can address this. And also neuroblasts migrate with really characteristic speeds, trajectories, and the directional persistence, which is very different from SVZ neuroblast migration. And also the chemorepulsion cues. I have mentioned a couple of times of chemorepulsion between the daughter cells and also between the two interacting cells. So we think this chemorepulsion might serve as a general mechanism to distribute neurons within hippocampus. And also, lastly, I told you experience really exert potent effects on in vivo migration dynamics of neuroblast. So enriched environment really promote dispersion of the neuroblast, and as a result of increased motility and exploration of the neuroblast. So I just want to spend the next 2 or 3 minutes and just tell you what's the bigger questions we want to ask next. So all this is really good, nice and good. But what we really want to ask is how all these environmental cues, those extrinsic stimuli, can influence local circuits and intrinsic factors to regulate neuronal migration. I'm not even go into the details of what the circuits we're manipulating, though I have some information here, but I think I'm running out of time. And I think we label one specific inputs here, and we inject retrovirus to label the neuroblast, and then we use a chemogenetic approach to activate this specific circuit component. Then here is what we got. So this is just at the baseline. Cells move very randomly. They're nice and happy there. And this is a little bit slow, but you see this CNO injection. That's the point we activated the circuits. Now it should be what you're paying attention to. So now you can really see all these cells forming a chain. They are moving one after the other. So they seem to know where the cues is, and we think that is the circuit activity, which serves as a cue to actually drive the cells to move into one direction. So we have all this quantification here for directional persistence. Their velocity do not seem to change during migration, but what changes is the directional persistence. And the next aspect we examine is like if this system is amenable for genetic manipulation. So we want to manipulate the intrinsic factors. So we just choose this gene DISC1 as a point of entry because this is the gene that involved in migration in the adult stage and also in the embryonic stage. So upon knocking down DISC1 gene, that if we record the cells for like 11 hours, they don't seem to actually move much. They pretty much stay in the same position. If we do a global quantification for that, and we actually see under control condition, this is 45% cells are moving, but under the DISC1 knockdown condition, the majority of cells are not migratory. And last, I just want to show you this video very quickly. So you can see this is the only cell actually we detect from DISC1 knockdown. This cell, the other cell just die. And this being said, this system is actually we can track the cell death as well because we have some characteristic criteria for cell death. If you haven't really seen the cell disappear quickly, but then here's another one for cell death. So the summary for the last few slides is the conjunction of microendoscopic imaging and the circuit manipulation use chemogenetics, and the gene manipulation use retroviral mediated single cell genetics knockdown approach really allow us to investigate the cross-link between extrinsic and intrinsic factors in regulation of experience-dependent, activity-dependent neuronal migration in the adult brain. So last but not the least. So since like you see a lot of movements of the cells in the brain, so the last question I want to ask you is your head itchy yet? But you don't have to answer it, just think about it. Last but not least, and I want to end my talk with thank all my lab members. So just as I pointed out earlier, so this system is developed by my graduate student, Andrew Crozer. He really actually single-handedly employed this system and get all the surgeries, get all the combinations to make it work in dental gels. And also my collaborator, Gary Stuber, who provide the initial technical support for us to continue the project, and the Inscopics, and Stephanie, and Vanya for invitation and coordination of this event, and my funding source and my lab. Thank you very much, and I can take some questions now.